Today we will be studying the second major process of protein synthesis, which is called as translation. So translation is the transfer of the transcribed or programmed information from the mRNA to a polypeptide chain. So in the first step, which is called as transcription, the information coded on DNA is copied or transcribed to a pre-mRNA, which is then spliced and modified to form the mature mRNA. The information coded on this mRNA is to be converted into the synthesis of a polypeptide chain or a protein. So during this, what exactly happens is that the 4-letter nucleotide language or the 4-letter nucleic acid language is translated to the 20-letter protein language. So what does this refer to? What does this 4-letter nucleic acid language refer to? Because mRNA is a ribonucleic acid which is made up of 4 types of nucleotides. They are adenine nucleotide, cytidine nucleotide, guanidine nucleotide and uridine nucleotide. So there are 4 types of nitrogen bases which make up the 4 different nucleotides. They are adenine, guanine, cytosine and uracil. So that is what is referred to as the 4-letter nucleic acid language in mRNA is translated to the 20-letter protein language. The 20-letter refers to the 20 biogenic amino acids which are used for the synthesis of different types of proteins. So there are 20 types of amino acids which are used for protein synthesis. So that is being referred to here as the 20-letter protein language. The mRNA contains the information which is used for sequentially incorporating amino acids one by one to make a final polypeptide chain. And it is this polypeptide chain which will attain the secondary, tertiary and quaternary structure to form the final protein. So initially it is always a polypeptide chain which is synthesized during gene translation. So in the polypeptide chain, the amino acids form the raw materials which are to be sequentially arranged just as what the information is given in the mRNA. Now we will go to the information recorded in the mRNA. mRNA contains the recipe or the instructions for correctly incorporating the amino acids one by one. So how does that recipe look like? That is called as a codon. Codon is a set of 3 nucleotides which codes for a specific amino acid. So genetic code is a complex coding system which consists of a series of coding units or codons, and the codon is a set of 3 nucleotides which codes for a specific amino acid. That is why it is called as a triplet codon. So here from this table we can see that the codon UUU on mRNA indicates that it is phenylalanine, the amino acid which is to be brought now. If the codon on mRNA is CUU, it is leucine which is to be incorporated. If the next codon in the mRNA is UAU, then it is tyrosine that is to be incorporated. So each codon, it will code for a particular amino acid. So a codon in an mRNA calls for the arrival of a corresponding amino acid. Out of these 64 codons, all the 61 codons which code for amino acids are known as sense codons, but there are 3 which do not code for amino acids. They have been shown to signal the stoppage or termination of protein synthesis. So the presence of any 3 of these codons calls for the termination or stoppage of protein synthesis. So they are called as termination codons or stop codons. So from this table we can see that UAA, UAG and UGA are the stop codons. So the presence of UAA or UAG or UGA on an mRNA calls for termination of protein synthesis. So just now we discussed about the recipe. Now we have to go to the ingredients or the raw materials. As it was already mentioned, the amino acids or the 20 biogenic amino acids are used for protein synthesis. But in what form are these amino acids used during protein synthesis? Before the initiation of gene translation, the amino acids are selected from the amino acid pool of the cytoplasm, and before they are assembled to proteins, they are to be activated or energized. How this is done? This is done by reacting them with ATP and with the help of the activating enzyme amino acyl tRNA synthase. So this reaction is called as amino acyl tRNA synthase reaction. Each selected amino acid taken from the amino acid pool of the cytoplasm will react with ATP and the enzyme. So this will form a complex of amino acid, ATP and the enzyme, which can now be called as the amino acyl adenylate synthetase complex. This enzyme bound amino acyl adenylate is the activated form of the amino acid. So the amino acid plus the enzyme plus ATP gives amino acyl adenylate synthetase, and this is also called as the amino acid AMP enzyme, from which pyrophosphate is removed. This amino acid is now transferred to a tRNA molecule, and then the AMP associated with the activated amino acid is also removed. So the amino acid will first react with ATP, then it will form a complex with the enzyme, in which process the pyrophosphate will be hydrolyzed, and then it will get associated with tRNA, during which AMP will be released. Now it is called as a charged amino acid. Now we will go to the function of tRNA. tRNA can be considered to be an intermediator or an adapter molecule which helps in positioning the amino acids in the exact position in a growing polypeptide chain. So it is a type of RNA molecule that will help in decoding an mRNA sequence into a protein. So here the tRNA has got a specific region, which in the figure you can see below as the anticodon loop. Before going to the various regions in tRNA, we will see how the codon, amino acid and tRNA are connected to each other. Each codon on an mRNA represents a particular amino acid, and each codon is recognized by a specific tRNA. Or every kind of amino acid, or for every kind of biogenic amino acid, there will be a specific tRNA. For example, here in this diagram you can see a tRNA which is meant for carrying leucine. Similarly, there will be tRNA which will carry only methionine. There will be tRNA which will be carrying only alanine. Thereby it is called as alanyl tRNA. So every tRNA is specialized to carry a particular amino acid. And in the diagram you can see this tRNA has assumed a clover leaf-like model, where below you can see a loop-like region which is called as the anticodon loop. So here in the diagram you can see in the anticodon loop of the tRNA, the sequence is GAG. So with the help of the anti-codon loop of the tRNA, it will come and bind with the corresponding codon on mRNA. So during protein synthesis, on mRNA there is a codon which reads as CUC. That means it is calling for the amino acid leucine. But this amino acid is to be brought by that tRNA which is specialized for bringing leucine. So it is this tRNA which is shown or which is marked as green in the diagram on the left, where you can see that it is having a charged amino acid, that is leucine. Now look at the anti-codon loop of the tRNA, where the sequence is GAG, that is it is complementary to the codon on the mRNA. So it is the mRNA that carries the codon for the amino acid leucine, but that tRNA which brings leucine has got a sequence complementary to this mRNA codon, and that region of tRNA which has got the sequence complementary to the codon on the mRNA, that region is called as anti-codon region. So during protein synthesis, when this tRNA brings its corresponding amino acid, it will be coming and binding with the mRNA using this anti-codon region. So the anti-codon on the tRNA will come and bind to the codon region, the corresponding codon region on the mRNA. It is called as codon-anti-codon pairing. So the codon CUC on mRNA calls for leucine amino acid, but it is this tRNA which brings leucine towards that area, and it is this tRNA which gets associated with the mRNA. That is why it is called as an adapter molecule, or it is a mediator. In the diagram on the right also, we can see that on mRNA there is the codon, and on tRNA there is the anti-codon loop which is quite enlarged in the picture in the right side. And in the anti-codon of the tRNA, there will be again 3 nucleotides. Just like the codon on mRNA is a triplet, the anti-codon on the tRNA will also be a triplet. And in that triplet, even if the last nucleotide wobbles or changes, it makes no difference in anti-codon-codon pairing. That is what is called as wobble hypothesis. That is how the tRNA acts as an adapter molecule for positioning the amino acids in the exact position in a growing polypeptide chain. Now, before going to the process known as translation, we will just have a checklist on the prior requirements for translation. So the first one is that mRNA which is carrying the codons for every amino acid to be incorporated into the new polypeptide chain. So mRNA carries the information, so it can be considered as the recipe. So that is the first requirement. Second is a site for protein synthesis. That is nothing other than the ribosomes. So ribosomes are the sites for protein synthesis. So the mRNA has to get associated with the ribosomes. Now the raw materials, the amino acids. And the amino acids are charged, and they are not coming singly, but they are coming associated with the corresponding tRNAs. So apart from these 3 requirements, there are also many other enzymes and proteins which will also facilitate the process. And translation also consists of 3 steps. The first step is called as initiation, the second is called as elongation, and the third one is called as termination. Initiation represents the start of peptide chain synthesis. Elongation represents the lengthening of the polypeptide chain, and termination is the end of peptide chain synthesis. So translation occurs on a polysome, that means an mRNA associated with several ribosomes. and it requires the participation of a variety of enzymes and protein factors, and GTP is being used as the source of energy. So we go to the first step, that is initiation. Initiation calls for the formation of a structure which is called as the initiation complex. There are two types of initiation complexes. The first one is the primary initiation complex, which later forms the secondary initiation complex. So we go to the primary complex. The primary complex is formed by the association of mRNA, as you can see on the left side of the diagram. The mRNA, which is marked as red. So that is the first component of the primary initiation complex. This mRNA now associates with the smaller ribosomal subunit. So that is the second member of the primary initiation complex. So first is mRNA, then the smaller ribosomal subunit, then the initiator tRNA. What is meant by initiator tRNA? Initiator tRNA is always that tRNA which brings methionine, the amino acid methionine. That is why it is called as initiator tRNA. So, methionine, the codon for methionine is AUG. It is considered to be the initiation codon. So, in any protein synthesis, AUG is the initiation codon which codes for the amino acid methionine. So, for the formation of this complex, first the mRNA has to get associated with the smaller subunit of the ribosome and then with the methionine tRNA or the initiator tRNA. Then, there are some proteins also associated with the process which are called as initiation factors. And finally, GTP. So, these are the components of the primary initiation complex. Now, on the right side of the diagram, you can see the secondary initiation complex. So, during this transition, the GTP is getting hydrolyzed to form GDP and energy is being released. And using this energy, the secondary initiation complex formation gets completed, during which we can see that the larger ribosomal subunit also associates with this structure. So, if we take the steps of initiation in a nutshell, binding of the smaller ribosomal subunit to the mRNA, then an initiator tRNA with the anticodon UAC base pairs with the start codon AUG on the mRNA, where in the left side you can see the codon anticodon pairing, that is AUG on the mRNA, the initiation codon, and UAC on the methionine tRNA. So that is called codon anticodon pairing. Now later the larger subunit completes the initiation complex and the energy molecule GTP provides energy for this assembly. In prokaryotes, instead of methionine, the initiator amino acid will always be formyl methionine. And so there the initiator tRNA is formyl methionine tRNA, whereas in eukaryotes it is methionine tRNA. Now we move to the second step, which is called as elongation. So in this diagram, before we proceed to elongation, in this diagram we have to see the three sites on the ribosomal subunit. The larger subunit of the ribosome, we can see three sites marked as A, P and E. A site is called as the acceptor site. P site is called as the peptidyl site and E site is called as the exit site. So what is A site? When the polypeptide chain synthesis continues, newer and newer amino acids will be getting added. So whenever a new amino acid comes or whenever a new tRNA comes with its associated amino acid, it will be occupying the A site. That is why it is called as the acceptor site. So the acceptor site is the binding site for all the incoming amino acyl tRNAs which are involved in the polypeptide chain synthesis. But the only exception is the initiator tRNA. The initiator tRNA will be binding to the P site, which is next to the A site. So, excepting the initiator tRNA, all the other incoming amino acyl tRNAs which are part of this protein polypeptide synthesis will be coming and first occupying the A site. So that is the function of the A site or the acceptor site or the amino acyl site. That is, it is the binding site for all incoming amino acyl tRNAs which are involved in the protein synthesis process. Now, second is the P site. It is in the P site that all incoming amino acids are added one by one to form a polymer. The amino acid polymer formation takes place in the P site. So P site, it is to the P site that the initiator tRNA binds. That is, except the initiator tRNA, all other amino acyl tRNAs occupy the acceptor site, but the initiator tRNA, it will be occupying the P site rather than the A site. Now, so we can consider the P site as the binding site for the initiator tRNA, and its second function is that all the peptidyl tRNAs with an attached peptide fragment will be seen on the P site. Consider in the P site there is the initiator tRNA with the amino acid methionine attached to it. Now the A site is empty now. Now we go to elongation. So elongation is the lengthening or polymerization of the peptide chain by the successive incorporation of more and more amino acids. For this process, certain protein factors are also required. They are called as the elongation factors. Elongation includes all the reactions from the formation of the first peptide bond up to the incorporation of the last amino acid, because we know that as each amino acid is getting added to the existing polypeptide chain, it is a peptide bond which is established. In the P site, let us assume that it is the initiator tRNA which is sitting with its amino acid methionine. Now in the elongation process, you can see that a new tRNA is coming with its corresponding amino acid. And where is it coming and binding to? It is coming and occupying the A site. So, the incoming amino acyl tRNA is now attached to the A site. Now, there are two tRNAs sitting together. One tRNA is having methionine, that is the initiator tRNA, and the second tRNA is carrying its corresponding amino acid. Now, these two amino acids are to be linked together by peptide bonding. There is peptidyl tRNA on the P site or the initiator tRNA on the P site, and there is a new amino acyl tRNA on the A site. So, during elongation, the methionine on the initiator tRNA is transferred to the amino acid which is located on the amino acyl tRNA which is there in the A site. So, from the tRNA which is now on the P site, the amino acid is transferred and peptide bonded to that amino acid which is now in the A site. So, this is called as transpeptidation. This peptide bonding is complete. So, this is called as transpeptidation, and this is mediated by the enzyme peptidyl transferase or peptide synthetase. Now, the tRNA on the P site has no amino acid attached on it. The amino acid has been transferred to the tRNA which is on the A site. Now, what happens? Now, the ribosome moves forward. You just imagine that the mRNA is sitting there itself, and imagine that the ribosome moves forward. So now when the ribosome moves one step forward, what happens? This tRNA, which is now on the P site, or the empty tRNA which is now on the P site, will now occupy the E site. And that tRNA which was now on the A site, or which is now on the A site, will migrate to the P site. So these tRNAs are not actually migrating, but it is the ribosome that moves forward. So what happens? The tRNA which is empty now will later shift to the E site, or it is translocated to the E site, and that tRNA which is having the peptide chain or the peptide bonded amino acids will now be on the P site. This is not accomplished by the movement of the tRNAs, but it is accomplished by the movement of the ribosome. So you just imagine that the mRNA is sitting there itself, the tRNAs are also sitting there itself, but it is the ribosome that moves forward, which will cause a rearrangement of seating of the tRNAs. In the final diagram, you can see that the empty tRNA, which is now in the E site or the exit site, is unloaded or expelled, and the tRNA which is having the peptide bonded amino acids are now in the P site. So which site is now free? A is free now. Now the next amino acid tRNA comes and it occupies the A site. With the help of the enzyme, the tRNA which is already having 2 amino acids bonded to it, it will transfer those 2 amino acids to the amino acid which is now present on the new incoming amino acid tRNA. So now the amino acid tRNA, the new amino acid tRNA will be having 3 amino acids incorporated to it. tRNA which is now on the P site will be empty. Now the ribosome again moves forward and the process is repeated. So to begin with, the initiated tRNA binds at the P site during initiation. Then the second incoming amino acid tRNA binds at the A site, which is the first step of elongation. Soon methionine from the former, that is methionine from the initiated tRNA which is now located in the P site, is transferred and peptide bonded to the latter amino acid residue. That means to the tRNA which is now located on the A site. As a result, we get a dipeptide, and this is synthesized by peptide synthetase or peptidyl transferase, and the process is called as transpeptidation. Now ribosome moves forward. As a result, the unloaded initiated tRNA gets pushed out from the P site, which then reaches the E site, and it is pushed out. At the same time, the second tRNA, which is now with the attached peptidyl residue, comes to occupy the P site, and the third amino acid tRNA comes in and occupies the A site, and the whole cycle of events is repeated, forming a tripeptide. In this manner, amino acids are added to the growing peptide chain one by one. So, A site is for every new incoming amino acid tRNA. P site is for the tRNA which has come just before this new amino acid tRNA, or the P site tRNA. The tRNA which is on the P site will be having the polypeptide chain which has grown so far, and E is the exit site. Now the last step is termination. Termination is the cessation of or the stoppage of peptide chain elongation and the subsequent release of the completed chain. So how is this governed? This is governed by a termination codon. So just as we mentioned before, the elongation process continues and finally an instance is reached when the ribosome moves forward and reaches the termination codon of the mRNA in such a way that the A site is now facing the termination codon or the stop codon. So, as elongation continues, the ribosome is moving forward along the mRNA and the ribosome moves forward and finally reaches the stop codon on the mRNA at a position where the A site is facing the stop codon. This leads to termination. So now what happens? Some proteins come into action. They are called as release factors. These release factors, they will recognize the termination codon, and these release factors will bind with the A site. So far what we were expecting at the A site, so far we were expecting new aminoacyl tRNAs into the A site. They will bring the corresponding amino acids with them. But when the codon is a stop codon, but if the A site is facing a stop codon, no tRNA will be coming. Instead, a release factor will be coming and binding with the A site. Now, these release factors, they will interact with the peptide synthetase and make it hydrolytic. So, what will happen? This peptide synthetase will hydrolyze the ester bond between the last tRNA and the last amino acid of the peptide chain. So what happens? The completed polypeptide chain is released. It is the tRNA, last tRNA is released free, and the completed polypeptide chain is also released from the polysome. The last tRNA gets expelled from the P site to the outside, and the ribosome also dissociates from the mRNA as subunits. These subunits can re-enter the ribosome cycle for the synthesis of another protein molecule. The detached tRNA or the expelled tRNA can pick up another amino acid and continue its activity. It is true with all the detached tRNAs, but the detached mRNA is hydrolyzed to ribonucleotide units by ribonucleases. And during elongation and even after termination, the polypeptide chain undergoes repeated folding to attain its characteristic secondary, tertiary structures. The new chain formed after amino acid synthesis will always have a terminus which is having methionine, because methionine was the initiator codon. Methionine was the first amino acid which is always incorporated. So before completion of the protein folding, the methionine is removed by enzymatic action. So these are the various steps involved in gene translation of protein synthesis.